Hi everyone, this is Miss Romani, and in this lesson, I will take you on a tour of the cell. Place a drop of pond water under a microscope, and you'll discover hundreds of single-celled organisms called protists swimming around. It's incredible. Just one tiny drop of water can contain all these living organisms. They are busy swimming, photosynthesizing, and eating each other. And each of these organisms consists of just a single cell. And that is because cells are the basic units of life. And a single cell can be a complete organism and perform all the functions that keep an organism alive. All living organisms are made up of cells. And so far, most of you have learned about cells that look like this representation of an animal cell, which is a great model to help us understand the structures within cells, and one that we will definitely work with. However, it is important to understand that not all cells look like this. The protists that I just showed you do not look like this, and even most animal cells don't look at all like this. A human body, for example, has different types of cells, like muscle cells, and brain cells, and fat cells, nerve cells, blood cells, each with a unique structure that determines their function. There are actually over 200 different types of cells in a human body, and that's just looking at a single species. The variety of cells is incredible when we take into account different types of organisms that exist. For example, bacteria cells. As you may recall from last year's biodiversity unit, bacteria are made up of a single cell, but a specific type of cell called a prokaryotic cell. The main characteristic of prokaryotic cells is that they don't have a nucleus or any membrane-bound organelles inside the cell. They have DNA, but it is organized into chromosomes that are free-floating and not encased in a nucleus. Every other cell, like animal and plant cells, for example, are called eukaryotic cells. Eukaryotic cells have a nucleus, as well as a variety of membrane-bound organelles and other cell structures, which is actually what will be the focus of today's lesson. And once again, let's focus on how structure determines function. So a brain is made up of nervous tissue, which is made up of nerve cells, which in turn is made up of specific structures and organelles that are necessary for their very specific function. So let's take a tour of the cell. And let's start with the endomembrane system. The endomembrane system is just a group of organelles and cell structures that tend to work together in order to basically make lipids or proteins that get packaged into vesicles within the cell. The reason why the endomembrane system structures will package either proteins or lipids is varied. A lot of them, and most of the time, they're packaged for secretion out of the cell so that they can be moved out of the cell. So these are lipids or proteins or sometimes carbohydrates that the cell is making but that are needed elsewhere. They can also be packaged so that they can be inserted into the cell membrane so that the packages essentially go to the cell membrane and then become part of the cell membrane. Or they can basically make special enzymes that are then put into these packages that stay within the cell and serve to either detoxify the cell or recycle old cell components, and they become special little structures, like for example lysosomes. The first component of the endomembrane system is the nucleus and the nucleolus inside of it. The nucleus holds and protects the cell's DNA. DNA does not leave the nucleus, but the genetic information coded in DNA is needed outside the nucleus in order to make proteins. So the nucleus is where DNA is read and copied or transcribed into a molecule called RNA. And RNA can then leave the nucleus through nuclear pores on the nuclear envelope. On the inside of the nucleus is also a dense structure called the nucleolus. The nucleolus is where the cell structures called ribosomes are made, and ribosomes are made from a combination of proteins and RNA. In general, eukaryotic cells are defined by the presence of the nucleus. However, there are exceptions. like the mature red blood cells that are found in mammals. Because they don't have any nuclei, mature red blood cells do not contain DNA and cannot make any RNA. Consequently, they cannot divide or make any proteins on their own, which is fine for red blood cells because they are made in the bone marrow of stem cells, and those stem cells do contain a nucleus. On the other hand, some cells, like muscle cells, can have more than one nucleus. Multiple nuclei mean multiple copies of genes, which are needed for production of the large amounts of protein and enzymes that are needed for muscle contraction. Attached to the nuclear membrane, we find the endoplasmic reticulum. The endoplasmic reticulum is basically a continuous membrane system that is mostly involved in the processing of molecules that are produced by the cell. It is divided into the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum has ribosomes attached to it. Ribosomes are in charge of protein synthesis, so the rough endoplasmic reticulum is involved in this process. It basically provides a place where some ribosomes in the cell can then make proteins, and then it helps with the folding and modifications of these proteins into their final structure. It also then packages the proteins it has processed into packages called vesicles that will then be transported to the Golgi body or Golgi apparatus. Now, the smooth endoplasmic reticulum does not have any ribosomes attached to it, so it is not involved in the manufacture of proteins at all. Instead, it is involved in a variety of other functions, starting with the manufacture or the synthesis of lipids, like for example, lipid hormones called steroids, as well as the synthesis of some carbohydrates. The smooth endoplasmic reticulum is also involved in the breakdown of drugs and toxins, a process called detoxification. And a third function is one of storage. Some cells in the body will actually use the smooth endoplasmic reticulum to store calcium ions, especially muscle and nerve cells, which use those calcium ions in order to function properly. So getting back then to the idea that forms determines function. Liver cells, which are responsible for detoxification of the body, have an impressive amount of smooth endoplasmic reticulum. And cells of the ovaries and testes, which produce the steroid hormones estrogen and testosterone, contain large amounts of smooth endoplasmic reticulum as well. Now, once the endoplasmic reticulum has modified the proteins or lipids, they are then sent to the Golgi body in a vesicle. Now, the Golgi body is sometimes called the Golgi apparatus or sometimes called the Golgi complex. And the Golgi apparatus is basically a series of membranes that are shaped like pancakes. And these single membranes that make up the Golgi body, and basically all the other membranes in the cell, are made up of a phospholipid bilayer, just like the plasma membrane. So the Golgi body basically absorbs vesicles from the rough endoplasmic reticulum, gathers the molecules inside them, and then combines them to make molecules that are more complex or that needed some finishing touches in order to complete them. It then takes those big molecules that it has modified, packages them up again into vesicles, and either stores them for later or sends them to the plasma membrane so that they can be excreted out of the cell. The Golgi body is also the organelle that builds lysosomes. Lysosomes are basically the cell digestion machines, and we will discuss those in a minute. And so once again, getting back to the idea that form determines function. Since the main function of the Golgi body is to package substances that are needed to be secreted out of the cell, cells in body systems that are basically in charge of secretion will need extra Golgi bodies. Like for example, cells in glands that produce either hormones or enzymes. Like for example, the pancreas that produces both hormones and enzymes. The pancreas produces two hormones, insulin and glucagon, and a whole bunch of enzymes that are responsible for digestion. Our brain and nerve cells are cells called neurons. And neurons basically produce little proteins called neurotransmitters that are used in order to transmit nerve signals from one neuron to another or from one neuron to a muscle, say, for example. And a third example are special cells called B cells that are part of our immune system. B cells produce little tiny proteins called antibodies. And these antibodies are basically secreted out of the B cells into the blood in order to help fight infections. And in case you didn't know, vesicles, as shown here, are just little membrane packages that mostly transport material throughout the cell, but sometimes also store them. Now, lysosomes are basically specialized vesicles that hold a variety of hydrolytic enzymes. These enzymes are proteins that are first created in the rough endoplasmic reticulum, are then packaged into vesicles, and sent to the Golgi apparatus. The Golgi then does its final work to create the digestive enzymes and then pinches off a lysosome. And from there, the lysosome will float in the cytoplasm until they are needed, like, for example, to break down invading bacteria or to digest some food, depending on the type of cell that it is. Also, it can be used to recycle old organelles, like, for example, this mitochondria right here. Sometimes the lysosomes are used to break down the cell itself, a process of cell suicide called apoptosis. So, what might trigger apoptosis? Well, there are a few reasons for a cell needing to self destruct. Like, for example, when a body part is no longer needed, like in tadpoles that no longer need their tails, or when the membranes between our fingers need to be removed during development. Another reason for self destruction is what happens when a cell becomes cancerous, or at least starts to become cancerous. That basically triggers a self destruct mechanism in the cell that essentially prevents tumors from forming. Cancer and tumors basically occur most of the time when too little apoptosis occurs. This results in malignant cells that will not die and will accumulate and will then go on to destroy body parts. Another organelle that is basically an enzyme package is a peroxisome. Peroxisomes are similar to lysosomes in that they contain enzymes, but peroxisomes contain oxidative enzymes. And oxidative enzymes are in charge of breaking down toxic compounds, like, for example, alcohol or hydrogen peroxide. So, if you know that the liver is in charge of detoxification of the body, you would then not be surprised to find out that liver cells contain peroxisomes because peroxisomes help detoxify the body. In particular, the liver contains peroxisomes in order to help detoxify alcohol and hydrogen peroxide. Now, vacuoles are also membrane packages. However, they do not move cell products around or contain enzymes. They basically serve a very simple storage role. They are there to store water or food or waste or fat. Plant cells tend to have one very large vacuole that is full of water, and that vacuole helps the plant cell maintain an internal pressure. Basically, plants use their cell walls to provide support, but the size of the cell may increase or decrease depending on how much water is present. So plant cells do not actually shrink because of changes in the amount of cytoplasm, but mostly because of changes in the amount of water that is in their vacuoles. When plant vacuoles are empty of water or when they lose a lot of water, the plant may become droopy or wilted. And when the plant gets watered again, the vacuoles will fill up again and the plant can regain its structure. Now, vacuoles can also play a special role in protists. These single-celled organisms live in freshwater environments and because of that are constantly taking up water. And so they have these special vacuoles called contractile vacuoles that essentially can contract and relax in order to pump any extra water out of the cell. And what this does is that it ensures that the cell does not end up too full of water, which can actually cause the cell to burst the cell does not find a way of getting rid of that extra water. And so contractile vacuoles basically are very important in these freshwater protists because it helps them from basically dying by bursting from too much water. And the final part of the endomembrane system is the ribosomes. Ribosomes are the protein builders or the protein synthesizers of the cell. They're like the construction guys who connect one amino acid at a time and build long chain polypeptides. Ribosomes basically come in two parts that assemble when they need to get to work in order to read the genetic code and build a protein. Both parts of a ribosome are made out of a combination of protein and RNA, and they're made in the nucleolus. Ribosomes are special because they are found in both prokaryotes and eukaryotic cells. And because, unlike any other cell structure we've covered so far, they are not surrounded by a membrane. And remember, prokaryotic cells cannot have any membrane bound organelles, but ribosomes do not have a membrane, so they are actually found within prokaryotic cells. Now, in eukaryotes, ribosomes can be found free floating in the cytoplasm, but also attached to the endoplasmic reticulum. Next is the mitochondria, the powerhouse of the cell. We will become well acquainted with the mitochondria during our next unit because the mitochondria's job is to produce energy for the cell in the form of a molecule called ATP. The mitochondria has two membranes, an outer membrane that is smooth and an inner membrane that has many folding called cristae that increase the surface area of the mitochondria. And most of the reactions involved in ATP or energy production are found in that inner membrane. So the more foldings, the more energy the mitochondria can produce for the cell. And it's not just the amount of cristae foldings that can differ between cells based on their energy needs, but also the number of mitochondria found in each cell. Cells that use a lot of energy, like for example our heart muscle, and pretty much all muscle, will have more mitochondria than average. Human heart muscle, for example, will have over 5,000 mitochondria per cell. This is a lot compared to the average of about 500 mitochondria per cell. And liver cells also have a lot more than average, but not as much as muscle cells, with about 2,000 or more mitochondria per liver cell. And another interesting thing about mitochondria is that they have their own DNA, which is found inside the interior of the mitochondria, a space called the matrix. This DNA that is found within mitochondria has 37 active genes. And there are active genes. They code for actual proteins that are needed in the mitochondria during cellular respiration. And so mitochondria even have their own ribosomes that are used to make the proteins coded for by this mitochondrial DNA. So essentially, mitochondria are like tiny little cells within our cells. And that's pretty much exactly what they are, tiny cells. There is a theory called the endosymbiosis theory that uses evidence, like for example, the fact that both mitochondria and chloroplasts have their own DNA, to hypothesize that eukaryotic cells actually evolved from prokaryotic cells that consumed but did not digest smaller prokaryotes. So perhaps some type of aerobic ones that used oxygen to make energy and then became our mitochondria, or some photosynthetic ones that make sugar, cheese, and light energy and became the chloroplasts that are found in algae and plant cells. Another neat thing about mitochondrial DNA is that because during fertilization, the egg provides all the organelles for the new baby, the inheritance of mitochondrial DNA is what we call matrilineal, meaning that we inherit them exclusively from our mothers, not our fathers. This is different than nuclear DNA, which we inherit equally from both parents. Both mom and dad each provide 50% of our nuclear DNA. But mitochondria DNA comes exclusively from our mother's side. And so our mitochondria is 100% exactly like our mother's mitochondria. and like our grandmothers and like our great-great-grandmothers. Any difference that there might be in mitochondrial DNA would be due to mutations that accumulate in the DNA over time. And mutations do happen, but not very often, less frequently than once every 100 people. So a person's mitochondrial DNA is probably identical to that of his or her direct maternal ancestor, probably about as many as a dozen generations back. And this fact can actually be used to connect people across decades. So, for example, if a particular type of mitochondrial DNA was found primarily in Africa, then we can conclude that anyone anywhere else in the world that would have that same type of mitochondrial DNA had an ancestor from Africa. So DNA test kits like 23andMe and other such tests actually use mitochondrial DNA to determine ancestry. Now, chloroplasts are another type of organelle that are kind of related or contrasted with mitochondria because they also have their own DNA. And chloroplast is the organelle that is responsible for photosynthesis and is found exclusively in plant or algae and pretty much any other photosynthetic eukaryotic organism. Chloroplasts contain the green pigment chlorophyll, which is used during photosynthesis to make sugars. And chloroplasts, like mitochondria, also have a double membrane. And they also have these little stacks of membranes inside that called thylakoids. And these are the areas that provide that extra surface area that is needed for capturing light into usable energy during photosynthesis. And all of these organelles are not simply floating freely within the cell. Instead, they are anchored down and held in place by a dynamic system of protein filaments that make up the cytoskeleton. The cytoskeleton gives the cell structure. They also help during cell division. For example, spindle fibers are part of the cytoskeleton. They also allow for movement of organelles within the cell, as seen in this animation of a vesicle being moved by a kinesin protein from one part of the cell to another. And it's basically moving along the protein fibers of the cytoskeleton. The cytoskeleton is also involved with cell movement. Whenever a cell needs to move, it's the cytoskeleton that's usually responsible for that. Like, for example, in the case of structures called cilia and flagella, which we'll discuss in a little bit more detail in a second. So there are three types of protein fibers that make up the cytoskeleton: microtubules, which are the thickest, microfilaments, which are the thinnest, and intermediate filaments, which are somewhere in between the two. Microtubules are what centrioles are made of. They are found only in animal cells and produce spindle fibers that move chromosomes around during cell division. And both cilia and flagella are also made of microtubules. Flagella are long, thick tails that whip around and sometimes twirl, pushing a cell forward. And cilia are little hairs that you might see on the outside of a paramecium or other protists, and they flap back and forth to help the cell move. Protein fibers are also found inside structures called pseudopods. Pseudopods are not permanent structures of a cell. They are temporary arm-like projections of a eukaryotic cell membrane. They are filled with cytoplasm, and pseudopods or pseudopodia primarily consist of actin microfilaments. They also may contain microtubules or intermediate filaments, but they're believed to be mostly made out of microfilaments. And the word pseudopod basically means false feet. It comes from the Greek words pseudos and podos, meaning false and feet respectively. And as such, they are often used for movement by some cells, and also for feeding or for taking in molecules from the outside of the cell. This amoeba, for example, is using pseudopods for moving around, but in this particular video, or very short video clip, it is using its pseudopods to feed on this paramecium. Now, the cytoplasm is the fluid that fills a cell and is found between the cell membrane and the nuclear membrane. The fluid in the nucleus is called the nucleoplasm and is separate from the cytoplasm. Each of those fluids have very different composition. The cell organelles are suspended in the liquid portion of the cytoplasm called the cytosol. And as we learned a few minutes ago, these organelles are held in place and moved around by the cytoskeleton. And finally, for this lesson, we have the cell wall. The cell wall is a structure made of sugars that surrounds the cell membrane. In plants, it is made out of cellulose, while in fungi, it is made out of chitin. And while the cell wall protects the cells and gives them shape, they also allow a plant to grow to great heights. And that's it for this lesson. We did cover a lot of different cell structures found in eukaryotic cells, but we did not cover one in particular. We did not cover the plasma membrane or the cell membrane. I will devote one lesson exclusively to the plasma membrane. So the next lesson will be just about that. So that's it for now, and I'll talk to you soon.